If you are worried you have Lyme disease, or just like the outdoors, and want the peace of mind of knowing whether you have Lyme disease or not, there is a new Lyme screening test based on decades of research by Dr. Richard Marconi, a professor at VCU Medical Center. For more information, visit glymedx.com. That's glymedx.com, or email at info@glymedx.com.
Infectious diseases, research, medicine, health. Welcome to Outbreak News Interviews. And now, broadcasting from the Outbreak News Skylar Studios in beautiful West Central Florida, here is your host, microbiologist and editor of OutbreakNewsToday.com, Robert Harriman.
As many of you know, I've been doing a series on the podcast titled Parasites 101. And I thought I'd like to do something similar with virology and viruses. And I reached out to someone who I clearly think is one of the best people to do this with, one of the best virologists and virology educators to help me out with this. So here is the first episode of Virology 101, and please welcome Professor of Microbiology and Immunology in the College of Physicians and Surgeons of Columbia University and all-around media guru, Vincent Racaniello, Ph.D. Dr. Racaniello, welcome back. It's been a while.
Hey, Robert. How you been?
I've been busy. How are you doing?
Staying busy as well.
I noticed. Okay, well, let me go ahead and start, and I want to get a little bit of get a little personal with you here. And I'm going back months ago, and you sent me this picture, and it's a car license plate, and it said viruses. And I still chuckle to today. And it clearly tells me that this is your passion. So what I want to know is how and why did you get into virology as a profession?
Well, I didn't always love viruses, I have to say. I guess I was in college, and I was looking for something to do. I majored in biology, but I ended up graduating without really a clear path in my career. So I went back home and I got a job in a microbiology lab, actually. And I worked at that job for about 8 months or so. And during that time, I realized I didn't know anything and I needed to go back to school. But I also read a book called Fever by John Fuller, which was, I think, a 1970s account of the first emerging infection, Lassa virus, which happened in Nigeria. Mm-hmm. And that totally sold me on viruses. I thought they were amazing, and I had to do it. So I decided then I wanted to go back to school and study virology, and that's really how it started. I mean, I'd always been interested in science, but the focus on viruses really came from that one book and that one account of an emerging infection.
Interesting. Now, let's get down to the meat and potatoes of all this. I want to go over some of the basics today. And Dr. Racaniello, what exactly is a virus? What makes a virus a virus?
Okay, so I've thought about this for many years, right? I have written virology books. I've taught lots of virology courses and blogs and podcasts. Here's my current definition of what is a virus. A virus is an infectious, obligate, intracellular parasite made up of some kind of genetic material. It could be DNA or RNA, and it's surrounded by a protein coat and sometimes also a membrane. Okay? Now, we can deconstruct that because it's a little complicated. Infectious is obvious. It can go from cell to cell or person to person or host to host. Obligate intracellular means that the virus has to get inside of a cell in order to reproduce. No ifs, ands, or buts. No way a virus can do anything until it gets inside a cell. And then the word parasite, as you are very familiar with, parasite takes something from the host. It harms the host in some way. So that's the definition. Infectious, obligate, intracellular parasite. The genome is DNA or RNA, which is unusual because most all other life on Earth we know is DNA-based. And then that genome is packaged in a protein coat and sometimes has a membrane on top of it.
Now, you said that this is your current definition, so has this evolved over time?
It has. It used to be more complicated and had more things in it, but as I've thought about it and we've written, different editions of our textbook and try to become clearer, we have reduced it. So I think this is about as minimalist as you can get.
Gotcha. Now, one thing that not everybody agrees on, and I've read differing views on this question, and I hope you can clear this up. Are viruses living organisms?
I thought about that a long time too, and that's a really interesting question. So the way I look at it is like this. A virus is an organism with two phases, all right? That's really important. There's the virus particle is one phase. So when people think of viruses, they think of these little particles that you can't see, but which can infect you. That's one phase. And the other phase is the infected cell. When the virus particle gets into a cell, takes over the cell to make more virus particles, that's the infected cell. So the way I see it, two phases. One of them, the virus particle, there's no way that's living. It's a virus particle made up of some DNA or RNA and protein. No way it's alive. It can't do anything. However, when it gets into a cell, the infected cell is certainly living. The virus has made itself part of the cell. It's taken over. So that phase is clearly living. So if you look at it that way, the particle, the infected cell, two phases of this virus organism. Virus particle, no way it's alive. Infected cell certainly is alive.
Let's talk a little bit about history. And I was wondering if you could spend a little time talking briefly about the discovery of viruses.
Well, the discovery of viruses, the early days, was driven by disease. In fact, the first virus discovered caused a disease of tobacco plants. It was called tobacco mosaic disease. Mm-hmm. And tobacco in the 1800s was already a big economic product. And there was this disease that made the leaves unsuitable for sale as tobacco. And people wanted to figure out what it was. Now, by the late 1800s, we already knew that bacteria could cause disease. And it turned out that this tobacco mosaic disease was not caused by a bacteria because the agent could pass through a filter that could retain bacteria. For the first time, people showed that if you, let's say, took a diseased tobacco leaf, you ground it up in buffer, you filtered it, the filtrate, when applied to new tobacco leaves, could cause the disease. The first time a filterable agent was shown to be able to cause disease. And that was very different from bacteria, which would be retained on the filter.
Right.
So they initially called these filterable agents. In 1892 and 1898, that virus was first discovered to cause tobacco mosaic virus. So that it was a really important discovery because it established this concept, first, that this agent is really small, and second, the agent itself couldn't replicate, couldn't reproduce unless you put it on a leaf. So that filtrate, unlike bacteria, would never multiply unless you put it on a new leaf. So 1892, 1898, that was the first virus discovered. And after that, of course, the concept took hold. First animal virus discovered 1898. That was foot and mouth disease virus. And then the first human virus, 1901, yellow fever virus. Mm-hmm. And all of these discovered because they caused diseases. And then there was a whole slew of viruses discovered in the next 30 years, rabies, smallpox, polio, bacteriophages in 1915, influenza virus only 1933. And I think this is interesting because nowadays we don't just discover viruses based on disease. We look for everything that's out there, all the viruses that are in the water and sewage and dirt, and we try and understand them that way. But early discovery was certainly driven by disease.
So influenza virus wasn't discovered till after the great epidemic of 1918?
Yeah, isn't that amazing?
Yes, it is.
That whole epidemic, we had no idea what was causing it. We knew it was contagious, obviously. We knew it was highly lethal, but we didn't know it was a virus until many years later.
Wow. Would have never guessed that. Now, the last question I have for you, Dr. Racaniello, is now how are viruses classified? It's like with bacteria or parasites, there's a genus and there's a species, but it's not quite the same for viruses, is it?
Well, we do have a little bit of that Linnaean classification, families and orders and genera and species, but and we use that to keep track of viruses, obviously. But classification, the basis for putting viruses in those families and genera and species has changed over the years. And initially, we used to look at a virus and say, okay, what is the nucleic acid? Is it DNA or RNA? What does the protein shell look like? Is there a membrane? How big is it? What kind of disease does it cause? And we would use that to say, this is an adenovirus, this is a poliovirus, this is an influenza virus. But nowadays, it's mostly sequence-based. We sequence the genome of the virus, and from that, we place it in a family, in a genus, species, etc. And this reclassification has resulted in some shuffling of viruses around. But for the most part, an influenza virus is an influenza virus. There just are more of them now because we're able to sequence. So we are pretty similar to bacteria and parasites in that they, too, now are based mainly on sequence analysis.
Yeah, true enough. Mm-hmm. Okay, I want to encourage the audience to check out Dr. Racaniello at Virology blog, and that's virology.ws. And he's got a whole bunch of different audio and video platforms he's doing, too, on virology and other microbiology topics. In fact, every time I go into the science and medicine section of Apple Podcast or iTunes, there I see TWIV or TWIM, so you're all over the place.
We got some new podcasts coming out soon, so keep an eye on that. And if your listeners would like a little more virology, my entire virology course here at Columbia University is online as videos. You can find them at virology.ws/course.
Excellent. Well, thank you, Dr. Vincent Racaniello, for your time and expertise. I look forward to
Thanks, Robert.
I look forward to talking to you again.
Thank you.